Professor, thanks again. So now we are 7 minutes late, which is almost okay, let's say. So we move fast to the next session, which is the session that we want to provide every year for dissemination of some European projects in nanomedicine. And this year we have a nice variety, even if we had to choose and to select a few of them, because there are many of them. And many new, like EuroNanoMedIII projects, for instance, etc. So we'll have first nTrack by Marc Masa. Then we'll have Safe and MedTech and TBMED2 OITBs that you all know probably. Then we'll have a very short update of the NOBEL project. I won't do the presentation, even if I'm not the coordinator, but Alex Superville will. And we will then go to ENAMEP, which is a new ENM3 project. So I will promote every speaker to be panelist, but we will start first by Marc Masa. Marc, hello.
Hello, good morning.
Good morning. So Marc is working at Leitat in Spain, and you will talk about nTrack, which is a multimodal nanoparticles for structural and functional tracking of stem cell therapy on muscle regeneration. So now you can share your screen, and Marc, the floor is yours for 8 minutes.
Okay. Thank you very much for the kind invitation. It's my pleasure to be here. And let's go to the point. I'm going to present this project. It's about just the nanoparticles for tracking the stem cells. So the idea is we want to solve the problem that these cell therapies are not cannot be tracked at the moment. So we want to develop a technology based on nanoparticles in order to be able to identify them and track and understand the viability and biodistribution of these stem cells once they are injected into the human being. So I'm sorry. The idea is to take advantage of the properties of these nanoparticles, which are gold shell, iron core nanoparticles, which allow us to use the different imaging systems available at the clinics. That means that we can run CT, MRI, and we have also modified these nanoparticles with radioactive isotopes to be able to use PET. The project, it's aimed to be ready for the first in man testing. So it's structured in a way it's to the role like a drug discovery project. So we want to we started from already having these nanoparticles, but the main goal here was to try to scale up under GMP conditions and then go to two different strategies. One of the toxicology approach, just to be sure that they're safe and they are not affecting any other cell functionality, and to understand the fate of these nanoparticles. One, these nanoparticles are loaded into the stem cells, and at the same time, we have to demonstrate that these stem cells and these are can be seen using these different imaging modalities. So, in a very simple way, this is the short summary of the proposal of the project. So we started with nanoparticles at lab scale. Then we shifted into the scalability for a large amount production, thinking already into the human setting, into the clinical trial in the future. And then we started setting up the protocols for the cell loading. Hopefully these nanoparticles, we'll show you later, are covered by glucose. That means the loading is quite easy. And then these cells are just characterized to be sure the viability and the properties of these stem cells are not affected. But the fact that these cells are loaded by the nanoparticles, we characterize the production of cytokines, and the key features of these stem cells remain, because at the end of the day, these cells are the ones who will provide therapeutic effect. Next, we split it into two in vivo settings. One, the small animals using mainly rat and mice, to start characterizing the toxicology profile, the biodistribution, and also the proof of concept for the efficacy. But also we used large animals. In this case, we used the sheep, that allows us to ease the translation into the preclinical level, into the human level, to the clinical level, because the anatomical structure of the hims and the joints of the sheep are quite similar to the human. And then we take advantage also, since we are collaborating in a hospital, we are running MRI and CTs of this sheep in the same systems that are being currently used in that hospital. So that means we have to collect the data in order to be ready to provide to the authorities the data required to reach according to the requirements they are asking for. At the same time, this data is being processed in a way to develop new artificial intelligence models that will also help to predict the potential migration of the stem cells into another locations that it's not desirable, or to understand the biodistribution based on this data that will be collected from the imaging data. We have two different, mainly one type of nanoparticles, as I said, magnetic core, gold set, and glucose coating, and this is the basis. And on the top of that, we add a radionucleotide to enable the PET imaging. So the two partners, the Bari University, as we started from their knowledge on the nanoparticles, and then we shifted into a German company who's able to produce a larger scale and GMP grade nanoparticles. This is non-radioactive, but this is the basis to start working with these nanoparticles. Then we take advantage also again for an Israeli company whose Pluristem is a leading company. And we, in order to avoid and to reduce the uncertainty, we take advantage of these cells which are already in clinical phase 3. That means that we don't have to take care, I would say, we'll have to consider any toxicological questions regarding stem cells since they are already in clinical. So it allows us to focus our research on the fact that we are loading nanoparticles into these cells that are already accepted and being used in a clinical setting. As I already advanced, the idea is to prove the use, how important, how relevant, and what kind of information is being provided in different animal settings and in different imaging technology for cell tracking. So that's why we use these two different animals. Actually, it was a requirement from the topic to use the large animals. That's why we used the sheep model for muscle regeneration, which is at the end of the day the idea that it's behind from the therapeutic point of view. Also, it's really relevant, and we took into consideration from the very beginning to involve people from the regulatory groups. And thanks, we have two different groups, one from Spain, another from the Netherlands, which has been involving from the very beginning, since we were in touch with the EMA in order to get some scientific advice, in order to say, okay, we know that it could be a new, different approach for cell tracking. So we need to know from the very beginning what kind of information, what kind of piece of information they will asking us for to progress into this project. And it's been really important since we had a meeting at the EMA at the very beginning, and now we are just moving into the National Competent Authority since it was defined as a medical device instead of a new drug that is really relevant in terms of development. And finally, we also considered that we will be running this non-GLP and GLP preclinical safety regulatory studies and toxicology in order to demonstrate that not only it's useful, not only it's allowing us to track them into using imaging systems, but also is safe for the use. Finally, the list the groups involved. It's, I would say it's quite balanced between academic groups and a small, medium enterprises and a large company. And just this is our guest that we've been used to, and from the, also we decided to have this kind of mascot as a representative of what we are doing. Thank you very much.
Thank you, Marc. We have time only for one question, if there is any. Maybe I have one. How and where do you do the characterization of all your nanoparticles?
The characterization of
the particles.
The particles we are doing in is being performed at the German company. They are doing characterization, but also at Leitat we're performing the characterization in order to know the size, the dispersity, and also the gold content and the iron content, in order to know if they're ready to be seen under a CT. And we have performing the phantom assays to know that the concentration it's enough to be seen when it's injected into the animals or in the human being. Final at the end of the day.
I'm sorry, I missed a question from Nicolas from the attendees. If you can ask a short question and I will have a short answer, please.
Yes,
please.
You mentioned the iron content for MRI imaging. Can the nanoparticles be imaged by MPI also?
Yeah.
Thank you. That was extremely fast. No, you can develop indeed if you wish.
No, just I'm sorry, I didn't catch your question. So what was the point you said about the iron of the nanoparticle? So the question was about
MPI, magnetic particle imaging.
No, I cannot answer this point. I have to just we just focus on MRI. I cannot tell you this point. I'm sorry.
Okay, but please keep in touch, and I'm sure you can discuss that privately.
Sure, thank you.
Okay, thanks a lot, Marc. Now we move quite fast because we are a bit late to Angel del Pozo. Angel, my dear Angel, please don't be late. Give me an 8-minute maximum presentation of these very important projects that we all know, which are the OITBs, the Open Innovation Testbed. And Angel will talk indeed about the project he's coordinating, which is SAFE and MedTech. Angel, the floor is yours.
Yeah, thank you, Alex. Thank you all for being here. I am just trying to find my presentation. We tried this morning and it worked, but now
okay, Angel, if you don't mind, I will skip your talk. You will be just after, and I will, because we are late, I will start now with TBMED, which is presented by
yeah,
Andrea.
sorry, I got it here.
Okay. You have
sorry for the suspense. Okay, can you see it properly?
Yes, please go ahead.
Okay, thank you very much for being here. Thank you for the organizers. Really sorry for not being in the nice Heraklion, where I think that there is some of the warmest people and the best food and the best also, of course, in the world. So I hope that we'll have a chance to be there next year or whenever. So I'm going to explain a little bit about SAFE and MedTech, what is the project and what are the main advances during, we are now entering our, we have accomplished our 18 months. So you know that it's time for us to reflect a little bit, to present our results to the Commission and to continue refining the project. Into SAFE and MedTech, we are trying to build, or we are building an innovative open access platform in order to offer companies and laboratories, spin-off startups, big companies, small companies, whoever is developing new medical technology based on nanotechnology. This is quite important, this distinction. We will try to offer them a know-how, network, services that they require to test and assess and upscale and further move to clinics and market their innovative technologies. As I said, in Safe and MedTech, the N stands for nanotechnology. We are proud, you saw yesterday that now specific projects on nanotechnology applied in medicine are being quite more difficult to build. We will stay here at least until 2023. So we hope that we can integrate the knowledge and the efforts that has been developed until now by the nanomedicine community in order to continue giving access to innovators to these services. So our consortium is huge, it's really huge, it's quite big. As you see there, we can start working at 8 a.m. and continue working till 8 a.m. but in South America, because we have 28 partners from 3 continents, mainly of course from Europe, but also from China and from Latin America, mainly in Colombia, because the call asked for recommended international cooperation, and of course we believe in the openness to the world that characterized the Horizon 2020. Into the consortium we have RTDs, SMEs, industries, governmental agencies, universities, clinical institutions. So we try to cover all the and integrate all the agents needed across the value chain of development for the medical technologies. In our case, as I said, for the nano-enabled medical technologies. So the people, you see them there. We as Biokeralty are dealing with the scientific and technical management of the project. We have people specialized in dissemination. We have people specialized in characterization, preclinical testing. I mean, we are many people there, regulatory, health technology assessment, clinical interface, business development. We are many people there, but as you can see, it's really a strong consortium. It's really, I would say that most of the relevant people in the nano technology applied in medicine and in medical technologies is represented there. So we are proud of that, and everyone is there giving a specific value for the project. I can hear somebody in the background. Please, could you mute your mics? Our mission, vision, and values are according to what I have explained. We envision that the nano-enabled medical technologies are here, are alive, and we want them to get into practice, to be fully developed, so we can improve the quality of life of our citizens, offering them new technology. People is not worried about technologies, worried about their health and the quality of life. And of course, to try to make this nano-enabled medical technologies a key economic driver for the sector, the medtech sector, and the European economy. Of course, we will try to help these talented companies with their nano-based innovations to the market. Based on that vision, our mission is to build a single entry point to provide these developers with an easy access to all the support that they may need in an integrated way, moving from the scattered offer where you go here and there, you find something to characterize, you find something for the in vitro, you find something for the in vivo, but you don't know the regulation. Even you have never heard about the health technology assessment aspects that at the end of the story will be crucial for your innovation to be reimbursed and adopted by the healthcare system, even private or public. So we are trying to build this single entry point in order to offer this easy access and support to the companies that are trying to develop and bring to the market these nano-enabled medical technologies. And for that, our values are, of course, the curiosity, the innovation and creativity, rigor. We are based on empirical verification and standard operational procedures to try to be effective. We foster transdisciplinarity, cooperation, and of course, we need to be strictly complying with the ethics and be transparent to create trust for our future customers. To do this, the project is built around several work packages. We have the first one is the initial evaluation and classification for the proposed medical technologies. This is building this single entry point, creating the interface with the clients and the customers. There are two really power work packages focus on, let's say, the more scientific and technical work, focus on physical and chemical characterization of these nanoparticles and nano-enabled medical technologies, coupled to the preclinical research in vivo and in vitro. We also include services for prototype and manufacturing and scaling up of these solutions. Of course, this is in continuous touch with the clinical interface, because some of the problems with nanotechnology sometimes is that maybe you do the preclinical research that you think that is right, or that the CRO proposed to you thinking that this is the right one. But when you move to the clinical evaluation, you find problems between your preclinical package and your clinical package. This, of course, driven under a continuous update on the regulatory landscape and regulatory roadmap, and also with what we call the Medical Technologies Innovation Hub, that tries to offer this innovator support also in the business development and the business opportunities, IP, etc., investors for their projects. As you see, this is not far from what was foreseen by the ETPN for these translation hubs. We also get inspired on this, and we will try to keep, let's say, keep the flame of all those initiatives into the project. We are not building a theoretical framework, of course. We are building a practical framework. So we have 4 test cases where we are applying this. They are companies that have joined the consortium. So we develop our framework, we develop our protocols, we develop what we think that the community needs. But in this case, it's good that we have these 4 different cases. One is hyperthermia based on iron oxide nanoparticles for tumors or other applications. This is proposed by an SME from RCL from the UK. We have Stryker, that is a big player in the field. So we have also a big company showing that this is interesting from them. They have developed an implant with a nano-structured surface to foster osseointegration and avoid bacterial contamination. We have also a company that is not into medical technologies, but they want to move to the sector. So they have a nanoparticle-based coating that avoid contamination and avoid fouling. They want to try if this can be applied in medical devices to avoid bacterial contamination and to foster a future remanufacturing or reuse of these devices. And we have finally a spin-off from the Technological University in Dublin, dealing with an advanced monitoring system for a continuous, let's say, online, inline monitoring of patients, for example, at intensive care units. Yeah.
I'm sorry, but you have less than 1 minute left. Okay,
it's perfect. Main achievements so far during these 18 months are the development of the interface to interact with the clients. We have, through the test cases, we are improving, trying to understand the needs of the developers. We have created a really powerful integrated matrix for the PCC and the clinical testing, combining the work packages 2 and 3. We have identified the needs for scale-up and manufacturing, and also the life cycle assessment and production, safety production and nanosafety. We are advancing providing the services for our 4 test cases. And we made an open call for COVID-19 related solutions together with the other open innovation testbed that has been, I could say, successful. So we will also provide our services for COVID-19, trying to contribute to this difficult situation. You see there how happy is the people with our project, and you see there how you can contact and communicate with us, and you have there my contact details. So thank you very much. Just lastly, Marc, we will promote another open call for all the nano-enabled medical technologies that are under development. This will be in March next year, but we will of course rely on Alex and ETPN to spread the news and other channels. But just for you to remind that there will be opportunity for developers to get our services for free.
Okay, thanks a lot, Angel. Unfortunately, we don't have time for any questions. I'm sorry, but you can contact Angel anytime. He's really quick in answering. Thank you again for having presented Safe and MedTech. We'll move to the next one. Can you stop sharing your screen, please?
Yeah.
Angel, thanks a lot. So the next one will be TBMED, another open innovation testbed. This one is an open innovation testbed for the development of high-risk medical devices, and it will be presented by Andrea Haiek from CIDETEC Spain. Hello, Andrea.
Hi, everyone. Thank you so much for having me. So, Alexander, are you going to put the pre-recording or should I do the presentation? Hello, my name is Andrea Haiek, and I would like to introduce you to TBMED, an open innovation testbed for the development of high-risk medical devices. TBMED actually comes as a solution to the challenges that several SMEs are facing. These are mainly due to two things. One is implementation of the medical device regulation, and the second one is the strict reimbursement approval process. They require not only to demonstrate the safety and performance of the device over the whole product lifecycle, but as well the benefits for the patient and the health system. Most SMEs are very small and do not have the knowledge, the contact, or the resources to comply with all the requirements by themselves, and therefore they are losing competitiveness. Here is where TBMED enters. TBMED is basically a connected network of labs that offer the possibility to access to all the required service using only a single entry point. And as important, offers a consultant that is going to accompany during the whole process in order to help you understand the requirements, how to fulfill them, and how to minimize the time and resources that you are investing. TBMED offers services that goes from a TRL 4, meaning the preclinical development, until a TRL 7, that is the clinical studies. It also includes several facilities that will help with the testing for in vitro and in vivo, as well as the prototyping, characterization, as well as the scale-up facilities. All of these supported by a strong quality by design methodology that will ensure robustness of the process and will help to understand the attributes that your product have and that should make them acceptable for the reimbursement. Moreover, TBMED also comes with an SME office that offers consultancy services on business development, market access, and possible financial aids. So the idea of TBMED is to have a holistic service that will help an SME from everything that they need from a TRL 4 to a TRL 7. In order to build this structure, 3 use cases has been used. The first of all, it's glycoban, that is an hydrogel tailored for oral bone reconstruction. The second one is a keratoprosthesis, that is a synthetic cornea. And the third one and final one is a hyperthermia treatment for cancer indication. It contains magnetic nanoparticles that are heated via an alternating magnetic field applicator. In the case of glycoban, there are 2 different indications. The first one is sinus lift augmentation. For this one, only glycoban is used, and it is on a more advanced TRL level. So it's about to enter into clinical proof of concept. Therefore, the services that has been offered are regarding counseling sessions with clinicians or with the health technology assessment in order to identify critical endpoints. As well, they have been helped with the documentation preparation for the clinical trial and the submission with the ethical committee and the agency. In the case of the second indication, this has as well a membrane that it's currently under development, and therefore services as a gap analysis to see which tests they will have to be performed and how to do the scale up of this membrane has been provided. And for both indications, a benchmarking analysis has been done by the SME office. In the case of the keratoprosthesis, the basic service that has been provided is optimization of the optic, together with the development of a quality control strategy. We have also set up the manufacturing process using quality by design concepts. And finally, we have had several counsel sessions with clinicians that make us to have our first in vivo test that is currently ongoing and is in rabbits. And finally, for the hyperthermia treatment, Several services has been provided, but the most important one has come from the counseling sessions with clinicians. The result of these counseling sessions was a change of the indication to one where they have more market and the device is much more appropriate. Also, it has been performed a market analysis to see which further applications or sectors they can expand after on. So right now we want to start expanding our services to companies outside the consortium in order to have some beta testers. In order to do so, we are opening 2 open calls during October and November. The first one is a so-called COVID open call, which was just closed last Sunday. Was directed mostly to SMEs which need advice regarding the process to how to perform a clinical proof of concept. We have received several applications and right now we are in the process of selecting the winners. In the case of the second open call, we call it the TBMED open call, and it's actually for companies that want to perform a clinical proof of concept or a multinational clinical investigation. As a requirement, they should have all the preclinical data available. This second open call will be launched in November, and the duration of the service will be more or less 2 years. Basically, any European company can apply to this call, even though it is primarily directed to highly innovative SMEs. TBMED as such will be directed to 4 types of companies: highly innovative SMEs, companies that need multi-center clinical trials or the reassessment of their marketed devices, companies that have already a marketed product outside Europe, but now they want to obtain a CE mark, and companies that want to expand into the medical devices sector. So with these 2 open calls, we want first to test these 2 client clusters in order to improve the service and later on expand to these other 2. And finally, I just want to show you who is actually behind TBMED. It's 13 entities around 4 countries in Europe, 5 research groups, 5 SMEs, 1 industry and 2 hospitals, all of them with significant track record in knowledge creation and innovation. And we all have joined forces just to guarantee a successful outcome. We actually hope that this initiative is of your interest. Thank you.
Thanks a lot, Andrea. I'm sorry, I need to switch between different audio devices. This is when there's a break of a couple of seconds. Great talk.
No problem.
Thank you so much. Do we have any question at this stage? I'm looking in the chat. So I have one. Well, OITBs are indeed very much needed to put some, let's say, great efforts together for not just for 1 or 2 or 3 years, but really to bring something different and something probably sustainable, hopefully sustainable in Europe. But my question is more on the client side, on the SMEs. Is it complicated, in your opinion, to let's say, to attract them, to explain the concept of an OITB because it's still new? Do you see any reluctancy, like for instance, for the IP management, these kind of classical things? How easy it is to convince the SMEs to come and benefit from your services?
I mean, I think the concept of an OITB, it's already starting to be sound one, but however, it's always easier to explain that it's a platform that will help like to manage all the services so they don't have to take care of the coordination between the labs. So I think it is important just to tell them that it's like the first contact point and then all the services can come just from one person. And then it's easier for them to understand the concept than just to say it's an OITB. Because in some cases it's not, as you say, it's not that easy to understand. Regarding the IP, well, it always comes the question of the NDAs and the contracts, but it's step by step. I mean, from the clients that we have been in contact, that probably we will have in the future some contracts. It has not been an issue at all. They do have questions, but once you define which is a process that they will follow and what kind of warranties they have, I think they are on board.
Okay, thank you so much. And good luck for the rest of the development and the new calls. ETPN will be at your side as we were to, let's say, do some promotion. These are very important And these are open services for the people to raise some successful business in medtech in Europe. So we'll do our best to support. Thanks again. Have a nice day.
Thank you, Alexander.
Okay, now we'll move to the next project that I know a little bit, which is the NOBEL project coordinated by ETPN. And hello, Alex, very nice background. I'm proud of you. So I will now broadcast the presentation of NOBEL, and Alex and I will be here for any question at the end. Thank you, Alex.
Thank you. First, I would like to thank you for having granted me with the opportunity to introduce the NOBEL project today. NOBEL aims to build a unique health tech ecosystem, paving the way for the health tech revolution in Europe. First, a quick recall. What are health technologies? Well, it refers to any technology, device, or digital solution designed to improve the health of patients. They are already used widely in diagnosis, monitoring, treatment, and rehabilitation. And yes, they are everywhere, from pacemakers to MRI, from simple glasses to advanced exoskeletons. Currently, a revolution is ongoing. Here, we all know well about new nanomedicine, enabling new diagnosis and therapy options. This revolution extends until augmented reality, allowing surgeon training, for instance, or new apps on smartphones, making patients actors of their own health. In NOBEL, we defend the vision that nothing is possible without a multi-technological approach. It aims to bring all technological domains with application in healthcare together. Nanomedicine, biomaterials, photonics, robotics, textile, and last but not least, data sciences. Here you can see what several technologies working together can create. An ambulatory exoskeleton controlled by the mind-thrown IA, allowing paraplegic people to walk again. Or devices reproducing all the aspects of an organ in a single chip. Moreover, health tech is a growing economy in Europe, as evidenced by these figures: a 115 billion euro market, 27% of the global market, top 1 patent application in Europe, plus 600,000 people employed in Europe, a singularity of the health tech ecosystem, 95% of the actors are SMEs. How NOBEL is acting to help them with the constitution of this single European health tech community. Here you can see our 3 axes of action: building the ecosystem, shaping the strategy, accelerating innovation. But how? We did organize and participate in a lot of events these past 3 years, as we do believe that it is essential to meet the people. Indeed, we are present in all the European important places, like ETPN2020 today, or the MedTech Forum, and so on. But also we have created useful dissemination channel for information exchange in the community. You can see that on our website. Please pay us a visit. Also, this is the purpose of all NOBEL organized events. For instance, each year we organize the Health Tech World Cancer Day, simultaneous educational event in all Europe, back to the World Cancer Day, to show how smart health tech help in the fight against cancer. On the other hand, NOBEL helps shaping a general strategy by publishing and broadcasting useful contents about the future of healthcare. Here are a few examples, but our main vision is illustrated here: the continuum of integrated care. It shows the shift from an acute phase-centered, responsive and reactive healthcare to a more preventive, continuous, patient-centered, value-based and data-driven way of practicing healthcare. This shift is allowed by the take-up of new health technology by all the actors of the healthcare value chain, from researchers to clinicians, for the final benefit of patients and society. It will help to avoid and reduce the acute phase, to improve rehabilitation, and globally, to improve the condition and the quality of life of patients. To help it further, we make available knowledge to help innovators anticipating the commercialization of their health tech, like this companion on successful translation, detailing testing, regulation, and reimbursement in Europe and beyond, open to download on our website. Or this video called The Innovation Loop, published by our partner Fondation Don't Know Qui. More importantly, Nobel actively helps to accelerate innovation by coordinating the Health Tech Translation Advisory Board, our custom mentoring service for the best health tech innovator in Europe, free of charge for its beneficiaries. Our core team of industry experts and entrepreneurs have developed for the past 5 years a unique culture of mentoring, as they act as a group with a complementary backgrounds and skills. You might have heard about Enatrans. During Nobel, there was a successful opening from the NanoMed tab to the Health Tech tab, which now supports projects of technology development from all technological domains that have applications in healthcare. Indeed, it has no proven value. As you can see here, more than 110 projects helped so far, raised more than 15 million euros. Here are a short list of the success stories. You might all know RubyNanoMen or Leaf Nano, for instance. Finally, I would like to introduce and thank to all our core partners, which are all experts with complementary background and networks. Here's a photo of the Nobel family. Our associated partners, who also contributed 6 nanomedicine national platforms coordinating their efforts. Here's the annual meeting. And other European technological platform with application in healthcare. Here's a photo of our last working meeting in Düsseldorf. To conclude, here might lie the heritage of the Nobel project. Driven by the ETPN, 8 technological platforms representing more than 4,000 technology providers in Europe have been brought together. Now, we are trying to build the largest community in terms of technology and stakeholder diversity, capable of delivering cross-tech solutions to address future healthcare needs. We are currently signing a memorandum of understanding to build a large health tech alliance, Health Tech for EU, which would be the voice of technology provider for the sake of patients. I would like to thank you for your attention. I am open to questions, and please visit our website, register, and send us a message if we can be of any help.
I agree with everything you said, Alex. Thank you for that. Nice presentation. Do we have any questions about the Nobel projects? I may just say that Nobel is now a survivor because it should have ended at the end of September, but we obtained a prolongation of 3 months just to benefit because of force majeure, indeed, because we were slow during the COVID-19 beginning of the crisis. And it's very good news for us because we will be able to do some things that were, let's say, blocked by the pandemics. And for instance, let's say that we will organize the Health Tech Award that will start very soon. And if you have an SME developing any nice technology in the health tech field, we will be glad to see you applying, because the prize pool will be very cool. And like, for instance, you will be the winners will be able to showcase their technology in plenary session of MedTech Forum next year, which all the key opinion leaders of the MedTech field will be here to attend. So we are really proud of that. Indeed, the Alliance, which is the, let's say, strategic heritage for Nobel. And finally, we fight hard to continue the tab after 6 years of efforts and of proven value. We have a brand new video for the tab. So if you want to go to the Health Tech tab or Nobel website, you can see it if you don't understand the value of the tab. But anyway, you will have a tab expert speaking at the end of the workshop today. Thanks a lot, Alex. That was really good.
Thank you.
And now to finish, I will ask to the last speaker, which is Ramaroson Andriantsitohaina from Université d'Angers. Are you here? Can you open your mic and your video, please?
Hello, can you hear me?
Absolutely, loud and clear. We cannot see you, but we can hear you. Now the floor is yours. You can share your screen for your presentation, please.
So I will just, where is my desktop?
Now we can see
desktop?
We can see cats now.
No, not easy cats, another, no. Do you have my?
Now we have it in presentation mode, but it's no big deal. You can go on. So you have 8 minutes. Thank you so much for being here. And you will present ENAMEP, Exosomes as innovative nanomedicine approaches to reverse obesity.
Thank you very much for choosing us to show, to give the quick look on what we are doing on the ENAMEP, which means Exosomes as innovative nanomedicine approach to reverse obesity and metabolic and psychotic complication with specific targeting of the hypothalamus. You know that obesity is a reaching pandemic proportion, causing 1,000 of deaths per year worldwide, directly or indirectly. Currently, there is no efficient treatment against obesity apart from the bariatric surgery. And most of the pharmacological strategies are associated with side effects and do not reach sufficient level of acceptability required by regulatory authorities. Strategy for the development of the successful treatment against obesity have been mainly focused on peripheral approaches, given the intrinsic complexity of the targeting of the brain. However, the growing knowledge of the hypothalamus mechanism controlling energy metabolism has made evidence that specific modulation of the neuronal circuit within the hypothalamus may offer a new, more effective target for drug development. That's why, sorry. Do you have it? I'm sorry. Do you have it? I have a problem.
You are back? No problem.
Yeah, I'm back. Good, thank you. Okay, so just the network is aimed to develop a nanomedicine approach to harness the biology of nanoscale exosomes as an innovative strategy to assess suitable tools for specific targeting the hypothalamic area, modulating energy balance. Indeed, exosome harboring genetic tools open a new way in the rational design of new strategy for the treatment of obesity. Now, how can I go far? So, the medical issue is that we use innovative exosome-based nanomedicine, which is an autologous cell therapy challenging the obesity-related disease. We will explore the property of exosome as a scaffold for the delivery of cargo of interest, yielding a novel therapeutic strategy. Controlling central regulation of obesity through this approach will spawn a definition of new strategy to offer with efficacy of the therapy. That this is presently the only way to target the hypothalamic area that can be delivered intravenously. This is important, making them affordable for therapeutic use, and the ultimate result will be the reasonable reduction of the cost of the management. Why are interesting for the solution? We know now that the ventral nucleus of the hypothalamus, called the VMH, plays a major role in the regulation of the energy expenditure by modulating and the modulation of the peripheral glucose and lipid metabolism, by controlling the thermogenesis in the brown adipose tissue through the sympathetic nervous system. We have already demonstrated that pharmacological inhibition of the hypothalamic AMPK leads to an increase of temperature, energy expenditure, and therefore a decrease of body weight. This is of important because several adjuncts with potential anti-obesity and or anti-diabetic effects act through the hypothalamic AMPK. Also, activation of the hypothalamic AMPK also mediates the pharmacological adverse effects of the worldwide used antipsychotic adjunct such as olanzapine, leading to oxygenase effect and glucose intolerance. Thus, it is crucial to reduce the impact of the adverse effect of this drug, which seems to be associated with AMPK activation. Finally, evidence has been provided of the interest of targeting the hypothalamic AMPK, including the NLRP3 inflammasome and the endoplasmic reticulum stress loop for drug development against obesity. Now, what is the solution that we are doing? We are going to produce nanoscale exosome able to target the central nervous system, in particular the VMH, without inducing any host-inflammatory reaction. How are we going to proceed? First is to do a neuronal targeted exosome. For this, we took immature dendritic cells in which we transfect with two proteins, LAMP2B, which is an exosomal marker, and the RVG, which has the property to cross the blood-brain barrier and to target the neuronal, and they can be transported in the retrograde fashion. When you transfect this immature dendritic cell, they will produce such an exosome with labeled LAMP2B and RVG plasmid. That will help us to reach the neuronal target. Now, how we'll do that, it will be specifically on the VMH. We take the advantage that the neuronal, the VMH has 90% controlled by the steroidogenic factor 1. So for that, we took our exosome and we loaded with a plasmid of interest, of the target of interest, AMPK, NLRP3, under the promoter of the steroidogenic factor 1 or SF1. This allows us to treat intravenously, for example, mice, obese mice as a model of obesity, and target the CNS and especially the VMH in order to increase thermogenesis through the bone adipose tissue, heat dissipation, and then feeding independent body weight loss. Why your solution is unique? Because this increases the specificity of drug delivery. That is one of the most important limiting factors with numerous actual therapy. This we limit host-immune reaction, we maximize the specificity, we have the aptitude to cross the blood-brain barrier to reach the brain and their efficiency to deliver the content that will be addressed either in vitro or in vivo, and the most importantly, that the ability of the intravenously treatment of the neuronal targeted exosome in the mouse model, leading to decrease of body weight. The safety of this exosome administration will less immunogenicity effect in relation with therapeutic dose used. Finally, in the end of the call, we will use human exosome from freshly monocyte-derived dendritic cell under good manufacturing practice-like condition to facilitate the translation to the clinical, though no clinical trial will be scheduled in the 3-year project. And ever TBMED, and probably there might be interest for TBMED within the next year. What is the level of the proof of evidence of our concept? We've already able to produce those exosomes just express LAMP2B RVG, as assessed here by the exosome markers LX1, TCG101, CD9, and CD81. When we labeled either control exosome or LAMP2B RVG exosome, and we looked at the biodistribution by intravenously treated mice, we see here within the hypothalamus an increase in the fluorescence of those exosomes bearing LAMP2B RVG, meaning that we are able to increase the proportion of the exosome within the VMH area. Now, to the specificity to target the VMH, we use the plasmid with the design target, in this case, the dominant negative of the alpha-1 subunit of the AMPK under the SF1 promoter. And those are the data. In the mice model of obesity, once we treated intravenously with 100 exosomes, you see the control and those treated by exosome with SF1, AMPK, alpha-1, alpha-1 dominant negative, you see a decrease in body within 3 days, and this lasts for 6 days, and I can tell you it can last for 28 days easily. And most importantly, this is not associated with no change in the food intake, and this is associated with an increase in energy expenditure. More importantly, when you look at the mechanism of the bone adipose tissue, looking at the temperature of the bone adipose tissue, you see here that when we treated the SF1 and AMPK dominant negative exosome mice intravenously, you have an increase in bone adipose tissue temperature, and this is associated with the increase of thermogenesis genes like UCP1, UCP3, PGC1 alpha, and PGC1 beta. So that is the proof of concept that we are able to produce neuronal targeted exosome loaded with target of interest and targeting exactly the VMH and we can use it by an intravenous injection to obtain the feeding independent body loss weight. Now, what is the next step? This is banned by 4 consortiums. So, as you can see, we have validated the dominant negative SF1 exosome in vitro, in vivo, and now we have to do it with other targets as NLRP3 or B. We investigate the effect of those exosomes on energy metabolism in diet-induced obesity, as I've shown you already previously. This is done by our lab and also our collaborator, Miguel Lopez from Santiago. And then this will help us to investigate the neuronal pathway that mediated the effect of this exosome. With regard to the adverse effect of antipsychotic agent, we'll investigate the potential of those exosomes to prevent olanzapine-induced metabolic disturbance, and this is in collaboration with Johan Ferno from Norway. And finally, as I told you, we try to going to obtain human exosome from freshly dendritic cell under GMP condition in order to translate it to the clinic. And this will be done by our colleague here in France with Marc Gregard in Nantes. So that will be, we hope that we will have the new way of treating obesity by this nanotechnology approach. I will end by showing our partner. We are 4 partners, myself in University of Angers, Miguel Lopez in the Santiago de Compostela, Johan Anders Ferno in Auckland University Hospital from Norway, and Marc Gregard in the INSERM in Nantes in France. And I would like to thank you, EuroNanoMedIII, for funding us, and I would thank you that you've chosen us to show our project. Thank you very much.
Thank you so much, Dr. Andriantsitohaina. That was really nice to finish by a new EuroNanoMedIII projects and not only H2020 projects or OITBs. So thanks again. Can you stop sharing your screen, please? Or maybe I can do it. No, I cannot do it. So you need to stop sharing, even if these cats are really cute.
Yeah.
You need to stop sharing right now, please.
Okay.
Okay, thank you so much. I think we have a question. Yes, absolutely, from Rosario. So the question is, thanks, great talk. Could you please comment how you treat the exosomes before in vivo administration, please?
How we treat exosome before, then there is no particular treatment of the exosome. Once we loaded the exosome with the SF1, with the plasmid of the target of interest under the SF1 promoter, there is no particular treatment. We just wash the exosome and we use 100 microgram per exosome that we intravenously injected in a PBS solution.
Okay, straightforward.
Something quite easy indeed. The most difficult part is the first part to build the assay to obtain this type of exosome. But after that, it's very easy to use. But I take the opportunity if there is some lab or SME we're interested, because our problem will be within 1 year or already now is to the upscaling of the production of the exosome. Yes, and toward the clinic. We can do it for small animals, but in the next step that will be, and I take the opportunity if there's someone interested in doing that.
We will help you in that opportunity. Please be sure that the ETPN can help you on this search for a partner for GMP manufacturing upscale. And just to let you know, so we have questions for you, so I will go fast. Ray Schifflers is asking what is the efficiency of plasmid loading in the exosomes?
Efficiency plasmid loading of the exosome. We, of course, we treat for the efficacy. We do in vitro study first on the specific GT1 7 cell line or the VMH cell line to see the decrease in phospho-ACC, for example, with regard to the MPKNS, to see the efficiency, and we calculate the efficiency of this. And we can inhibit by about 50 or 70% of the activity of the MPKNS. And this in vitro and also in vivo after the treatment, we have a slice of the hypothalamus and look at the activity of the phospho-ACC.
Okay, so you have several inputs on this efficiency. And last but not least, we have Flavia Castro saying thanks and great work. Can you comment the biological stability of these exosomes? And this will be the last question.
Okay, the stability of the exosome, indeed, this is it is quite stable for this exosome. We can last, for example, we can use it by 1 or 2 months when you put it on at -4 degrees. And we have already done a treatment, 1 month treatment, and the decrease in bodies is still working. And when we do the washout, and we still get after 4 weeks after this difference between the control and the decrease of the body with what change in food intake. And that is quite amazing. And we have all the parameters, decrease of adiposity and all involved in that. And all the, and this is really quite amazing.
Okay, thank you once again. That was really great to have you here. And thank you for the people asking questions. That was really nice. This is the end of this session for dissemination of European project. I would just let you know that on the ETPN website, there's a dedicated page for these projects. Some of them are already in this page, but the page will be updated with new projects like ENAMEP, for instance. And it's really practical because, for instance, you have a one-pager and so one-pager, let's say, a description of this project. But more importantly, you also have a single concatenated Twitter feed of all these projects. So if you want to get some fresh news from this project, this is the one-stop shop information you are looking for. Thanks to all the speakers. That was really good to have a European project, even if in the virtual meeting. So now we will have a short break, if you don't mind. Okay, let's have some fun. We will start back at 11:22.